this is the last talk of this morning. We're going to have the Professor Joe Thornton to talk about architecture and the evolution of epistatic interaction across 500 events.
Only that?
Good. So thank you very much for the invitation. I'm going to talk, I'm an experimentalist, so I'm going to talk about data, or as we say in Chicago, I'm going to talk about data. This is work done by Yanwu and Ben. The first half of the talk is published. Second half is totally new, and hopefully the presentation won't be too rough, but in spirit. So we're interested in epistasis and evolution, the causes of epistasis and the consequences of it for evolution. So the framework here is, as we've heard during Rama's talk, protein phenotypes are determined both by the first order context independent contributions of the amino acids that make up a sequence, plus pairwise and higher order effects. We define each of these effects as the average deviation of the set of sequences that have a sequence state or a combination of sequence states from the global average. We call this reference-free analysis, and it's analogous to a Potts model, essentially. So that tells us, that approach lets us parameterize the architecture of a protein and the phenotype that its sequence produces by the states that are in the protein. Each mutation that could occur in the protein then replaces the first order effect of some ancestral residue with a derived residue. But it also changes the phenotype by changing all of the epistatic interactions that that site has with all of the other sites in the protein and the amino acids that they happen to have at that moment. So a mutation incorporates first, second, and higher order effects.
So can I just, so Xij, that's the contribution of the combination of a mutation on site I and
The beta is the effect on the phenotype.
Yeah.
And the X is essentially the dummy variable that says this state, it's either 0 or 1. 1 if that state or combination is present in that sequence.
But I guess what is it Xij is Xi times Xj?
It's one if the pair
Okay.
I and J is present in the sequence. You can make it a product.
Okay.
1 times 1 is 1. Either 1 is 0.
Thank you.
It's 0.
What is the average over? It's
Over. Okay, so the intercept is the global average phenotype of all variants.
Okay, so you're taking a family.
The global average, and then this is the average of the subset that have that state. So it's basically ANOVA for sequences with respect to phenotypes. So in terms of an evolutionary process of substitutions accumulating across lineages, each substitution has the potential to change the effects of many mutations. You can have a cumulative effect of many substitutions, which will, in total, change the effect of introducing any future mutation and may even alter the epistatic architecture. That means the set of couplings and their values between sites. So this could affect evolutionary accessibility and the prediction of the phenotype that would be associated with any genotype. So let's consider some mutation which I've labeled as number 1, which is initially inaccessible because it's deleterious, it reduces the phenotype. But if there's a permissive modifier, number 2, which as drawn here is neutral, it could epistatically modify the effect of 1, making it advantageous, opening the door for that to occur during evolution. The converse could also happen where a modifier turns a mutation that was advantageous into a deleterious one, closing that path. So this means if this is the case, then evolutionary paths would be contingent on the sequence substitutions that happen to occur across a lineage, and our ability to predict the effect of a mutation would depend on the background. So this is something that could happen. We would like to know the extent to which it actually does happen. So we need some data on the nature and impacts of epistasis during the process of historical evolution. And here are the questions that we would like to know. During the course of historical evolution, to what extent do the effects of mutations change? What are the temporal dynamics of this process? Is it gradual or episodic? Is it directional? What kinds of interactions in the protein's architecture cause epistatic evolutionary change and the effects of mutations? And then at the higher level, does the epistatic architecture, the couplings and the values of those couplings, actually change over time? And what are the consequences of this for evolutionary accessibility?
When said this model, there's just one phenotype for the protein.
Yes.
One number. Okay.
Yes. So how are we going to get this kind of data? The strategy that we've been using is to combine deep mutational scanning with ancestral protein reconstruction. So deep mutational scanning is the preparation of large libraries of variants of some focal protein, such as every single possible amino acid mutation at each site in the protein, or every pair of mutations in some protein. And if we could do this using reconstructed ancestral proteins along a phylogenetic lineage, then for each of those proteins, we could determine how over time its effect on the phenotype changes. And we could learn something about the temporal dynamics of epistatic change. If we could do double deep mutational scanning or triple deep mutational scanning, we would get some idea of the couplings that define the protein's architecture and understand how and why substitutions along each lineage cause these modifications. So that's the strategy. We want to know the extent, the temporal dynamics, and the causes. So what's ancestral protein reconstruction? Very briefly, this is a phylogenetic method using Bayesian approaches to determine the posterior probability distribution of sequences at some ancestral node. In brief, in a cartoon example, given some set of extant sequences that are aligned, we can infer the phylogeny that relates them to each other. And if we are interested in the sequence at some ancestral node, the method proceeds site by site. It assumes that sites are independent. And given an evolutionary model of the, it's a continuous chain Markov model that gives you that specifies the probability of changing from any one amino acid I to any other amino acid J. We can calculate the probability of the entire alignment given the ancestral state, the tree, and the model. That's the likelihood of some ancestral state X. And using a Bayesian approach, we can calculate the posterior probability of any of the 20 possible amino acid states at that node. The maximum a posteriori state is the one that has the highest probability of giving rise to all of the sequence data that we see in the world today, given the phylogeny in the model.
Just so I'm clear, this part ignores epistasis.
It does ignore epistasis, yes. And let's talk about the consequences of that later. We've done a bunch of work on that. And so then the maximum likelihood sequence is the string of maximum likelihood or maximum a posteriori ancestral states. And because this is a posterior probability distribution, there are other plausible reconstructions typically, because not every site is typically going to be reconstructed with posterior probability of 1.0. But given that we can synthesize cDNAs that code for these reconstructed ancestral sequences, characterize them in the laboratory using the same methods we would use to characterize extant proteins. We can identify causal mutations for changes in function that happened during history. We can study the biophysical mechanisms by doing X-ray crystallography, for example, on the ancestral proteins, and we can do deep mutational scanning.
So how does one know the probability of switching from each amino acid to each other? Does that depend on the protein, or how do you
Of
That's
switching along a branch, you mean?
No, so for probability of mutating from from each amino acid to every other one is a parameter that would come into to.
Yes, that's embedded in the model, and there are models that have been fit to huge databases and phylogenies of many different protein families. There are family-specific models. They can be parameterized on a site-by-site basis. This is the generic version. Yeah. So I don't want to spend too much time here because it's not really the point. What I want to get across to you is that it's possible to reconstruct estimated ancestral sequences at any node on a phylogeny. And the more densely sampled and the better conserved the sequences are, the higher the confidence in the ancestral proteins and the trajectory of change across.
So just a clarification. So you say here there's a change in function. So now it's a different phenotype that's going to be measured, or it's the same phenotype when you mean a quantitative?
In what I'm going to talk to you about, it's just one phenotype. Yeah. So the system that I'm going to talk to you about today is steroid hormone receptor DNA binding domains. So these are transcription factors that have multiple domains. One is a ligand binding domain that recognizes hormones and upon binding specific hormones, changes conformation and has the capacity to activate transcription. And a DNA binding domain, which recognizes specific sequences and binds to them, specific DNA sequences in the flanking regions of target genes. So this multi-domain protein as a whole can activate transcription of target genes in a very specific way when exposed to their favorite hormone. I'm going to talk to you about estrogen receptors mostly. So this shows you the structure of one DNA binding domain on the DNA. These things bind as dimers, but I'm showing you here only the monomer. It's 76 amino acids long, and it's quite nicely conserved. It has very strong phylogenetic signal. So we inferred the phylogeny of the steroid receptor family from several hundred extant sequences. A cartoon version of the phylogeny is shown here, and I'm going to talk to you about two phylogenetic trajectories shown here in purple and green. There's a split here. They all have the same DNA binding function in our experiments. So I'm going to depict this big family here, this whole complex phylogeny for the rest of this talk, just in terms of the two lineages that are shown here and 9 nodes on the tree, 2 extant, and 7 ancestral. And the idea is to do deep mutational scanning in each of those ancestral backgrounds to trace changes in mutation effects along these phylogenetic trajectories. This is about half a billion years of evolution from this ancestor to each of the extants. So it's actually 1 billion years of evolution.
Sorry, can you repeat again? What is the function you're assaying? It's binding what?
Is binding to DNA.
Just the affinity.
Just DNA.
Yeah,
Specific?
I'll show you the assay and then ask the question again if you need to.
Sorry, how
So-
big is this protein? Like, how many
The-
amino acids?
The DNA binding domain is 76 amino acids.
And
so
that's
it's
the only
quite
part
compact.
you're reconstructing, just the binding domain?
Yes. The reconstruction has quite high confidence. This is a histogram of posterior probabilities across maximum posterior probabilities across sites. Mean of 0.96. The vast majority of sites have no ambiguity whatsoever. That's for this ancestor that I'm going to talk about numerous times, the ancestral steroid receptor, and then here are the other ancestors, similar high confidence in the ancestral reconstruction. All right, so here's the experimental approach. Make a library that codes for all 19 possible amino acid mutations at each site in the protein. Do that in all 9 of these backgrounds. So Yanmu did this work and it was quite immaculate. There are a little over 1,400 mutants per DBD. There are a total of 9 backgrounds for about 13,000 variants. They're all present in the master library. Clone that library into an expression vector, transform it into yeast with a GFP reporter. If it folds and binds DNA effectively, all of these bind so-called estrogen response elements. If they do this effectively, they activate GFP. They activate GFP in proportion to occupancy of the folded and bound state. Use fluorescence activated cell sorting to separate the library of cells according to their fluorescence. Sequence those bins, which allows us for each variant to estimate its mean fluorescence from its representation in each of the bins. Okay, so we then have the mean phenotype of all 1,400 some variants in each library. Okay, very high replicability across replicates. This is done in 5 replicates. I'm showing you 2 of the replicates here. So what this does is it lets us take for any variant in the library, its phenotype relative to the wild type. That's its change in function, and then across any interval of time, we can calculate the change in function. That is the change in the effect of the mutation.
You've shown all these being negative, so you've reconstructed one which is a local maximum.
I think it's the next slide will answer this. Oh, do I not have it? Okay, so all of the ancestors, I don't have this panel. All of the ancestors have nearly identical function, which is right about here. And this is for a given mutant that in this background, that mutation is very deleterious. In this background, it's slightly deleterious. In this background, it's slightly advantageous with respect to function. Okay?
Oh, it is slightly advantageous.
Yeah.
And is there some assumption that the ERE domain thing that did not change over this billion years? That the function was to bind that piece of DNA.
All of the wild types. Yeah, this is the same panel that I'm sorry that I'm missing. All of the wild types at each of those nodes binds the ERE sequence, which is AGG TCA palindrome,
And that's the same.
at almost identical levels. Okay. So first question I want to talk to you about is how widespread was epistatic change across the course of DBD evolution? And there is a wide variety of extensive epistatic change among the different mutations. So here's one that changes a lot. Here's one that changes very little. So we want a way of summarizing this. The first one I'm going to show you is the relationship between the maximum effect of the mutation on any background along the phylogeny and the minimum.
Sorry, I guess I'm lost about scale. What does 1 mean?
So these are arbitrary units of fluorescence.
Oh, they're just raw fluorescence or log fluorescence or?
It's linear. It's raw. Yeah. So to test whether the effect of each mutation differs significantly across the 9 backgrounds, we used ANOVA with an FDR of 0.1 to determine how many change over time. And here's what we see. 61% of the 1,400 mutants change significantly in 1 or more intervals. Of these, 22% change in sign. 39% don't change in sign, but they change in magnitude. Of those that do not change, The vast majority of them are simply completely destructive irrespective of background. And there's a small fraction that have no sign of significant epistasis, but are not unconditionally destructive.
Are there some that are essentially always neutral or not on this? So there's not a lot that are right by zero.
No, they could be. Ask the question again, please.
Are there some which are essentially always neutral?
Yeah, I mean, no, that's these. These are close to neutral all the time. These are totally destructive, no function left at all.
Okay, zero is up there, yeah.
Of background. Yeah. So the effects of most mutations, about 60%, change across the course of long-term evolution. So pervasive epistasis. There were epistatic changes in every interval, including the shortest one, which has only 2 substitutions in it. And if we plot the number of mutations that have significant changes in effect versus the length of the branch, the number of differences on that branch, you see a monotonic increase, which corresponds to every single substitution changing the effects of, on average, about 60 target mutations. So what's causing this epistatic change? Does it show signs of selection or drift? By selection, could these epistatic modifications reflect selection for robustness to mutation or evolvability or some such thing? So there's no evidence for any directional selection on changes in mutation effect. The mean is almost exactly 0. If we look at the percent of mutations that are deleterious or completely destructive, that does not change over time. So there's no apparent change in the protein's mutational robustness over time. Furthermore, if we plot the total epistatic change versus the sequence divergence, this is similar to what I showed you before. There's a nearly linear increase indicating that epistatic deviations in function increase gradually as substitutions accrue along the trajectory. So this looks like a process of epistatic drift. So are these changes gradual or episodic? And this can tell us something about the underlying architecture or hint at the underlying architecture. So we used classical approaches in evolutionary biology, where the effect of each mutation is treated as a trait, and it can evolve probabilistically along a trajectory, and we can fit different models to the process of change.
Can I ask a question? How much does the fact that you reconstructed the ancestors without epistasis shape the statistics you say on the last slide? Because I feel like it might be that if you get the reconstructions wrong, you systematically see things like this, but I don't have good intuition for that.
So it's a super interesting question, because I'm telling you there's lots of epistasis, and then I do the reconstruction without epistasis.
And the epistasis is drifting, and so is that because you just didn't fit it in the first place in the ancestor reconstruction?
So we have a paper in Molecular Biology and Evolution in 2025, which looks at this protein family and several other protein families and asks if we incorporate empirically fit degrees of epistasis, does that change the ancestral reconstruction? And the answer is no. And the reason for that is that the reconstruction is dominated by the phylogenetic signal that these branches all have a Y at this position. And so the ancestor has a Y.
It's just conserved enough that you don't have to worry about
Yeah, exactly.
So here's where it's 500 million rather than 3 billion. Relevant.
Yes, and it's a well-conserved protein. So if you try to do this with a protein that's noise all over the place, then the model will probably matter more, and model violation will probably matter more. And that's actually the take-home message of that paper that I was mentioning. All right, so we're going to compare 2 different models for the temporal dynamics of change. One is Brownian motion, that the effect of a mutation changes gradually and at random without direction, which implies that there are many causal factors. So it would look like this, and that would arise if the changes in an interval are being chosen from from a single distribution with constant variance. And that implies a model in which there are many causes with a range of strengths, which are being sampled at random across the trajectory. The other model is one of episodic change, where there's relative stasis punctuated by rare and large changes, which cannot be explained by sampling from a distribution with one variance, and would then and so this is modeled by having a mixture model with multiple variances, and that would imply rare changes of large effect. So we used a likelihood ratio test to test for the ability to reject the Brownian model on every single mutation. And we found that we could not reject Brownian motion for 92% of the mutations. We could for 8%. And I've just shown you two examples each. Here is Brownian motion with very large deviations. So the deviations are being sampled from a high variance distribution, as if this is a mutation that's very sensitive to modification by epistatic modifiers. Here's one that's also gradual, but with smaller degrees of change. Here's episodic, stasis, large changes. Here's episodic, stasis, small change, but that cannot be explained by the distribution of change across the other intervals. All right, so from all these data, we think we're seeing a process of epistatic drift, that there's a dense network of epistatic interaction in the protein that causes mutations effect to drift gradually and without bias during evolution as the protein accumulates substitutions. There are a small number of large effect couplings which are likely responsible for those episodic changes. Yes.
Sorry, I'm a little confused. You said you only have some number of sites, 76, you said?
76 sites, 19
There's
mutations.
a lot of conservation. So don't you kind of know how many, like under your model, it's just the substitutions that cause any changes. So you kind of know that there must have been, I don't know, between these at most 6 or something like that.
But remember, these branches have anywhere between 2 and 23 substitutions on each branch.
2 and twin, okay.
Yeah, that's a good question. All right, so the dynamics of this process look like gradual small modification epistatic drift, where each site has a characteristic epistatic sensitivity, but mostly lots of modifiers, as I said. Last thing from this experiment that I want to tell you about is insight into how quickly did mutations' effects become unpredictable because of this process of epistatic modification by substitutions. So what we did was we took all the mutations and we binned them by their rate of change across the phylogeny. And then we asked at each point on, at each pair of nodes along the phylogeny, given the sequence divergence between them, how decorrelated have the effects in one background become from the effects in the other background. So this shows you for 3 different categories, the decorrelation that happens across the entire length of the phylogeny from this deep ancestor to one of the extant ones. So here's one that gets decorrelated, almost completely decorrelated, moderate decorrelation, little decorrelation. And we can do that at each point along the phylogeny, and we can plot sequence divergence versus decorrelation and ask, what's the memory half-life? What is the degree of sequence divergence at which a mutation is half decorrelated from its ancestral effect? Okay, so this is basically the rate at which its memory of the ancestral effect is lost. So here's the distribution of memory half-lives among all 1,400 some. About 26% have a very short half-life. meaning it takes, they are half or more decorrelated across the length of this phylogeny, across this 500 million years. There's a large set, another 20% that have a medium half-life, that they're partially decorrelated across that long interval. And then there are about slightly over half that are essentially not changing. The shortest memory life half-life is just 3 to 9% divergence, meaning it takes just 2 to 6 substitutions to decorrelate it by half. So these are the most epistatically sensitive. We think they're likely to be the most richly coupled and by number of connections and magnitude of the effects of connections.
Just my lack of understanding about these DNA binding domains. So how many of the residues are essentially in direct contact with
You're about to see that.
DNA?
You're about to see it. Yeah, so about half of the mutations are fully or partially randomized over the course of the evolution of this domain. So this indicates pervasive historical contingency. The effects of many mutations became decoupled from their initial effects because of epistatic interactions with historical substitutions that occurred along the phylogeny.
Sorry, is this more consistent with the kind of local epistasis picture you say, or a more global epistasis picture? Or have you thought about that?
So yes, have thought about that a lot. So all of these backgrounds have nearly identical expression level and function. So we don't think it's the case that the impact of the mutation depends on the phenotype of the background, which is the sort of the model of global epistasis. So in this data set, that doesn't seem to be explaining this. These seem to be specific interactions. So the last thing I want to clarify is what's the underlying process of these substitutions that are happening? So we asked for the subset of substitutions, the subset of all mutations that actually happen on the phylogeny. What was their effect on the phenotype in the interval when they happened? And so gray shows you the effect on the phenotype of all mutations, and red is the substitutions. And you can see that there's a pretty sharp cutoff, slightly below 0, indicating a threshold, what's likely to be a threshold selection model, and that the DNA binding domains evolved primarily under purifying selection, that these substitutions that are accumulating are essentially neutral with respect to the phenotype, but they're highly consequential with respect to epistatic modification of other mutations. What's the impact of this epistatic drift on the fate of mutations? So here I'm going to show you for the 3 categories of memory half-life, 2 different plots. One is the percent of substitutions that were initially accessible in the deepest ancestor, but the path closed at one point or another, that the effect of the mutation went below that threshold. And for those with a short memory, virtually all of them, their path was closed at some point. This shows you the opposite. In the deepest ancestor, they were initially inaccessible, the door was closed, but then they became either neutral or advantageous later, also a large fraction of them in proportion to their memory half-life.
Sorry, but the absolute number, the distributions of delta F is pretty similar across short, medium, and long. Like this is percentage, but if I looked at the absolute number, I'm just wondering if the distributions are
like the long ones are mostly those ones that are always neutral or always
yes, that's what I yes,
totally destructive.
Yeah.
And the short and medium ones are fluctuating between neutral and sort of bad or a little bit good. Yeah. All right. So there are these windows of accessibility that are transiently opening and closing for, it's actually hundreds of mutations, because it's about half of 1,400. So several hundred of possible mutations are historically contingent because of this epistatic modification. And the extent of contingency, the extent of epistatic modification differs among substitutions. All right, so part two.
So just if you, out of this 1,400,
Yes.
how many of them do you infer that somewhere along the sequence existed, somewhere along the phylogeny existed? So I'm not
That
asking.
those mutations occurred.
Yeah.
Yeah,
They ever occurred.
That's this. These are the ones that occurred. So how many are there? There's about, on those two branches, there's, I think, 54. You're going to see it on a slide coming up.
I see, okay. But if you take the many branches to all the different extent animals,
Other ones on the phylogeny? I don't know. That's a really interesting question. We should
that would
do
sort
that.
of give a sense, right, of this as well, as if there are ones that never occurred.
Yeah.
How they would-
Yeah. Okay. So the second thing I want to show you is the complete double mutant scan. So we see this epistatic drift. What kind of couplings are causing it? What are the structural correlates of these couplings and so on? And does the architecture itself change over time? Do the substitutions cause couplings to change? So to do that, we're going to make a library of all singles, which we already made, and all possible double mutations. So all pairs of sites, all 19 times 19 mutations at each pair of sites. This is a big experiment. So we didn't do it in all 9. We did it in 3 backgrounds, the deep ancestor and 2 extents. And here's the number of substitutions. It's 57 that occurred along these branches. We got all the singles. We got 90% of the doubles in the library. Same experiment, 5 replicates. So here's how I'm going to describe to you the extent of epistatic coupling of individual mutations as well as pairs of mutations. We're going to use a modifier target framework. So any one mutation, we can call it I, can be considered a modifier. It has some effect on the phenotype, but it provides a background in which we can compare the effect of another mutation, J. What is J's effect on the phenotype when it occurs in the wild type versus when it occurs in the background of the modifier? So this shows you the effect of mutation J when it occurs in the wild type background. That's the expected effect of J on the background of I if there's no epistasis. But suppose that on the background of I, mutation J giving genotype IJ gives you this phenotype. Then there's epistasis. And we can calculate the epistasis term as the difference between the effect of the mutation in the wild type background versus the modified effect in the background of I. Okay? So I is the modifier, J is the target. For I, we can calculate its impact on all of the possible targets, all of the other mutations. And then we can repeat this for all the modifiers and ask how strong is each modifier across all targets. So it's a macroscopic view. Okay? I already showed you that. Okay, so now we have global epistasis here because we know there's a nonlinearity in fluorescence. There's lower bound, there's upper bound where the protein is unfolded, where the machine can't measure any higher or any lower. It appears from the distribution of sequences that it's a nice sigmoidal relationship, as you would expect for a simple model like this. And so we use a sigmoid transformation to transform fluorescence into the latent linear variable that's the effect of each state and combination. We censored any pair where any of the genotypes was outside the dynamic range of measurement or there was a high standard error. So we really want a high degree of precision in doing this. So we ended up with about, I think it's about a quarter of the possible pairs, but there's so many of them. There's 800,000 of them in the library that that we still have a lot of data to work with. All right. So for a modifier, we can express its degree of epistatic modification in two ways. This one is with the R squared, the degree of decorrelation for the effect of J on the wild type background versus the effect of J on the modifier background. Yeah. And the R squared, which is disattenuated for noise, shows you that this is a modifier that's fairly weak, but it does have some, it does cause some decorrelation in the effects of all targets. There are others that are quite strong, as you would expect. Okay, so that's the R squared. Yes, sir.
So any mutation could be a modifier or a target.
And they're all both, and it's
Okay,
in fact a symmetrical effect. Yep, that's right. Two other metrics we can use. We can take each of these, we can take the epistasis terms, the coupling, and we can ask about the distribution of the coupling for any I across all of its J's. We can ask about the mean of it and the standard deviation. And the one that's well correlated has a small standard deviation. The one that's less well correlated and has stronger epistatic modifier obviously has a higher standard deviation. Okay, so those are the metrics we're going to use. And we're going to ask, what does this look like across all of the modifiers? Okay. So here's the distribution of R squared across all 1,400 some modifiers. The vast majority of them are weak, with a mean of about, I think it's 0.93.
So this is the width of the distribution of the mean?
This is the R square. The effect of a mutation on the wild type background versus the background of the modifier. So how much does this modifier decorrelate the effect of all the other substitutions that could occur in the background of it versus in the wild type? So do I scramble everybody a lot? Do I have hardly any effect on everybody else?
It's just the slopes of the plots you were showing on the last.
Yep.
I guess I would take a lot of convincing as to why R squared was a good measure.
Well, maybe you'll like the sigma better. So the sigma is the standard deviation of the epistatic effects. And you can see that there are small number with a relatively high standard deviation, but most of them are small. And to give you an idea of scale, that mean represents about 1/10 of the dynamic range of measurement. So these are really pretty small effects for the most part, but there are some modifiers that are decorrelating their lots of targets by a fairly substantial amount. But most of it is weak, as we predicted based on that Brownian motion thing. Slight negative bias. Most modifiers are weak, and to the extent that they're modifying, they have a tendency to make other mutations worse, but not by a whole lot. And finally, here's the distribution of epistatic terms, the beta IJs. They're concentrated very close to zero, though this line is a normal distribution with the same standard deviation. So this actually matches a double exponential decay. If any of you math people can explain why that might arise, come find me, because I'm interested in this. But the phenomenon itself says most mutations are very weak modifiers. But it's heterogeneous. There are some strong ones out there. Okay. So the structure question. What's the cause of this heterogeneity? Why do mutations differ in their degree of overall epistatic coupling? There's a fairly coherent picture. So this is colored by that standard deviation of modification. You can see that where red are the strongest modifiers. These are mostly buried in the core, involved in the interactions mostly between the helices that are situating, that are the core of the fold and situating the recognition helix in the major groove.
Sorry, can I just ask a technical question about the experiment? So you swap in the DNA binding domain into the native yeast steroid thing that has the dimerization domain and everything else there, or these domains will just dimerize without the rest of the protein?
They dimerize on their own, and it's a reporter construct where they're fused to a yeast GAL4 activation domain. So we don't need the hormones, and they work nicely in yeast. Yeah. And so this shows you the correlation between the standard deviation and the relative solvent exposed surface area. And it shows a really quite nice relationship. It doesn't explain all the variants, but I didn't write it down, but it's a good R-square. It's by far the biggest correlation that we were able to find. So densely interacting residues are more likely to be strongly epistatically coupled. And if we plot the distribution of the magnitude of the individual epistatic couplings versus the distance between two residues, you can see that the strong interactions along the edges are enriched for close local interactions. There's still a few that are surprisingly distant, and I don't know how to explain those, but most of these are quite local. And because these are beta carbon distances, a lot of these are really very close, either next to each other or facing each other, and so on. What about the difference between substitutions and mutations? So before I showed you how the effect of substitutions differs from that of mutations, reflecting purifying selection. But this is how epistatic are the substitutions that occur during history. Gray are all mutations, red are substitutions. These are weaker epistatic modifiers than the set of all possible mutations. So evolution is enriching for weak interactions and depleting for strong interactions. So why is that? So you might be thinking, okay, substitutions are weaker modifiers. Why is that? Is epistasis somehow selected against? Is this another selection for robustness thing, something like that? I contend that this is a correlated response to purifying selection on the effects of the mutations themselves. Purifying selection takes out deleterious mutations. Deleterious mutations also have the strongest epistatic effects. So this shows you the median of the effect. The squares are the substitutions. The gray dots are all possible mutations. I've plotted the effect of the mutation on the phenotype versus its strength as an epistatic modifier. And you can see that those that are deleterious have the strongest epistatic effects, both negative and positive. Whereas the substitutions, this is the median, and they're truncated. There are not deleterious ones, except for that weird one, which I don't know how to explain. And by purifying selection, truncating these out, it truncates out those that are strong epistatic modifiers. Yeah, I have one more. So the weakness of historical epistasis is a correlated byproduct of purifying selection on main effects. Selection does not see epistasis except under rare circumstances, and it doesn't see it here. So this directly corroborates the idea of gradual epistatic drift due to many weak modifiers accumulating across phylogenetic trajectories. All right, so let me get to the one last thing that I want to show you, which is how does epistatic architecture change over time? So do these couplings and do the strength of modifiers, is that maintained across this long period of evolution? So what I've drawn here is showing you the distribution of R squareds for the modifiers in the 3 different backgrounds. It's identical. So no change in that. Here's the distribution of the strength of epistatic modifiers, the sigma, virtually identical. So at this macroscopic level, there's no change across 500 million years times 2. But if we look at the microscopic level, we see a different picture. So here I've shown you the sigma of individual sites. So the average sigma, actually this is of each modifier in the ancestral background versus the extant background. And there's some degree of decorrelation. So the overall distribution is the same, but some are getting stronger and some are getting weaker. And then this thing is the distribution of the epistatic couplings themselves in the ancestral background versus the derived background. And I'm going to show you this distribution. So some modifiers change in strength. This is what this looks like. Okay, so what should we see here? Most interactions are weak, the yellow stuff close to 0, and they stay weak. So all this non-epistatic stuff stays that way. But there's a small fraction of stuff where it starts out neutral and it becomes deleterious, or it starts out deleterious and it becomes neutral, and a little bit of stuff on the positive side. And there's like an equilibrium here, where the exchange for negative interactions from neutral is almost exactly counteracted by changes of other ones in the same direction or in the opposite direction. So why is this? It has to be because of higher order epistasis. If pairwise couplings change over time, it's because those substitutions are involved in higher order interactions with the pairs. But I want you to see that the change in pairs, this is the difference in effect across backgrounds, is considerably smaller than the change in first order effects. So there is this drift in couplings, but it's smaller than the drift in mutation effects per se. All right, so I'll go to my conclusions. Here's what I'll say. A protein's macroscopic epistatic architecture is defined by its fold. How much stuff is buried, how many residues are in contact with other residues. Over time, purifying selection maintains that fold, and the macroscopic description of epistasis doesn't change. But its microscopic architecture, exactly which mutation is coupled to which other mutation, and the value of that interaction is defined by the particular states that are at each site, and a substitution at a third site can change the value of an interaction between 2 amino acids. Evolution under purifying selection is typically thought of in molecular evolution as being essentially inconsequential. You have chance states coming in and out, they have no effect on the phenotype. And that's right. But they do have a major effect on the potential accumulation of other substitutions and on the underlying particular microscopic epistatic architecture of the protein. So the cumulative effect here is epistatic drift in the effect of mutations, as well as epistatic drift in epistasis. Epistatic couplings themselves drift over time as substitutions accumulate. So the take-home message here is that the effect of a mutation on a protein and its fate in the future or the past can be predicted or extrapolated into the future or into the past or into a homologue, but only if the memory length, the epistatic sensitivity, is longer or weaker than the time scale of the comparison. And this is also true of epistasis. So if you quantify epistasis in one background, some of those couplings will be maintained over time, but others will be sensitive. And the more time that passes, the more those couplings will change. I'll stop there.
So we're almost at lunchtime. Maybe you can take just one question. Okay.
If the question here is whether or not things that adrift are actually neutral, or whether they could be changes in the other parts of the protein which have a weak effect on this part and sort of push it between two sequences that looks at neutral under these measurements, or where it's so neutral that we don't have to worry at all about selection. Which would you think is more realistic?
So you have two scenarios. Will you clarify them?
One of
for
them
me?
is the population genetics one is it's just really neutral.
Neutral
It's really neutral.
and inconsequential for other mutations as well.
The other one is that there are always pushing and pullings on what's slightly good or slightly bad in this domain coming from the rest of the protein, other things interacting with the rest of the protein and so on. So they're quite different pictures of what some of the epistasis comes from, even though they're sort of not clear how you would distinguish them, I think.
Yeah. So I think there's good evidence for the model that changes that are truly neutral or nearly neutral can have strong epistatic effects. So I don't think you need like fluctuating strength of selection to make way for this sort of epistatic modification. But there's also the sort of thing that I think you're talking about, that there can be epistasis at higher levels, which creates a kind of systems drift, which lets some mutations occur neutrally that might not have been neutral at other times. So the expression of the protein goes up. So a mutation that compromises activity a little bit is less consequential, for example. So there could be that sort of thing happening over time, which lets some mutations behave as neutral and then modify the effects of other mutations. I think there's a lot of this sort of thing going on in molecular evolution. Yeah.
Let's thank the speaker again, and